make sure?
Yep.
Okay, great. Yeah, so I just want to say, first of all, it's nice to chat with you all, meet you all virtually, and thank you, Jessica, for organizing this. It's really exciting for me to get a chance to talk to folks out there. You guys are doing the real work, and so it's just really nice to talk to you because unless you stop by the site, I don't really see a lot of people out here in the field, so it's nice to chat with you online. And today, I'm going to talk to you about a project that we got funded through the LCCMR, this Measuring Pollen and Seed Dispersal for Prairie Fragment Connectivity, and it's in collaboration with Dave Moeller and Allison Shaw, both at the University of Minnesota. And so this is largely the first full year of this project, and a lot of the work requires a lot of time to process samples, and so it's only mostly there's really not going to be no results, unfortunately, but I am going to tell you about the work we're doing and show you some pictures of the field and the plants we're working on, so you can get an idea of what the results might be in the months, hopefully, to come. So in summary, our project aims to measure the prairie plant movement in the Minnesota region in order to determine habitat connectivity, which can support precision prairie conservation efforts here in Minnesota, and is especially important for pollinators because we really want to try to get an idea of how pollinators are using the landscape. And right, so as I mentioned, it's only the first year, so I won't really be able to give you results, but we will be talking about the project in general. Okay, there we go. Couldn't figure out how to move the screen. So as I'm sure all of you are aware, Minnesota has lost a significant proportion of its native prairie habitat. This is a vegetation map I'm showing here from the Prairie Conservation Plan, and in yellow shows the extent of the native prairie since 1908, which was about 18 million acres. And in red, as of 2011, we see the extent of the current native prairie habitat, only about 235,000 acres. So not only have we lost a significant proportion of our native prairies, but the way the habitat is arranged on the landscape has also changed. And this can really affect dispersal and movement. So not only it's no longer a large continuous swath of prairie, right, in the western part of the state, but instead the fragments that remain vary in size and shape and also distance from each other. So in the past, species could move widely across prairie habitats because they existed in these large swaths. But today, however, conservationists know very little actually about how current prairie species can move between these fragments. And so when I'm talking about connectivity today, I'm going to be thinking about it from the plant's perspective. We often think about, can different animals move between fragments, but because they're easier to think about animals moving because they move and they walk around. But today, I'm just going to think about how plants actually move. So with our project, we are at the beginning of understanding if prairie fragments in Minnesota are able to maintain connectivity for plants. And so if we promote the connectivity, determining connectivity of prairie fragments is important for conserving natural resources in Minnesota. First of all, connected landscapes are higher in quality for many organisms like pollinators or other insects, and even for plants. Prairie fragments are connected by when prairie fragments are connected by movement, this increases both plant genetic and species diversity naturally, which is important for other organisms in the ecosystem that rely on these plants. For example, pollinators rely on plants for pollen and nectar, and many native bee species specialize on only a few species of plants. So when pollinators move to new fragments, they need to have their host plants, or the plants they use for resources to be there in order for them to be able to survive. So if we promote connectivity of plants, we will also promote healthy communities for animals. And second, by understanding connectivity, we can help develop precision prairie plans. So for example, if someone wanted to restore a prairie on their land and we weren't sure they weren't sure where to do it, using connectivity metrics that we're going to be developing, we could determine where a new restoration should be placed in order to promote connectivity between existing landscapes. And this type of precision prairie restoration will allow residents and land managers to efficiently conserve Minnesota prairies. And so this is just a reminder that when we have healthy prairies for plants, we also have healthy prairies for insects. And I like this pollinator picture. So, okay, so let's think a little bit about how plants move. So to understand prairie connectivity from a plant's perspective, the first thing we have to do is figure out how far plants move. And plants do this in two ways. First, they move by pollen and seed. And so pollen is transferred from the father to the mother plant by either wind or pollinators, and this is called the pollen dispersal distance. So you can see here I have a depiction of two plants, right? And the pollinator is moving the pollen from the father to the mother plant. But then there's also seed dispersal, which is when seeds mature on the mother plant and then they disperse also by various mechanisms such as wind or other animals, and they land some distance away and then they grow. So we're working on trying to understand for several target species how these prairie plants move by pollen and seed. And so we're doing this work in Clay County at Bluestem Prairie. So our goal in finding a site was to try to find a prairie with large swaths of contiguous, just a large remnant prairie. So we can try to capture the maximum dispersal distance because we need to know where the sources came from. So if the prairie had been seeded, we wouldn't be able to use it. And so we're working at Bluestem Prairie. I'm sure many of you are familiar with it. It's kind of one of the flagship prairies, I think, in the state. And our big question that we're trying to answer is how far do pollen and seed move for common prairie plant species? And we want to know if this dispersal depends on the dispersal mode of the plant. So you can again have pollen that moves by either wind or insects over here, and you can have seed that either moves by wind or animals eating seeds and excreting them or sticking to fur, etc. And so for the first year of the project, we focused mainly on 3 species that moved by pollen by pollinators and seeds by wind. And these are the species that we focused on in the first year. And this, by the way, was last year. So last year, we focused on Rotibita columnifera, which is a species that is relatively short-lived, colonizes early, and seems to rely heavily on seed for maintaining itself in the community. And then we worked on Solidago rigida, which is a species that also colonizes easily from seed, but it has plume seeds like a dandelion, so it will likely travel a greater distance than seeds of for example, Rotibita columnifera that has more of a sunflower-like seed. And what I really am excited about for Solidago rigida is that it's clonal, but not massively clonal like other Solidagos, so it tends to stay in clumps. And so we'll be able to get an idea of vegetative spread as well. And then finally, Echinacea angustifolia, which is a similar dispersal mode, but is a later establishing species and longer lived. So we have a diversity of species that we worked on last year. And so what we did to work on this project is we went to Bluestem Prairie and we collected tissue from all of the potential fathers. So I'm going to be focusing mostly on pollen dispersal right now, but so basically we collected tissue from all of the flowering plants of these 3 species at Bluestem Prairie. And so you can see this is the 3 of us last year. I had 2 wonderful technicians, Jordan Prusensky and Dylan Jorgensen, and we worked all summer to try to collect tissue from every single individual and map them. And so what we're doing is we're collecting tissue from all the possible dads, right? All of the pollinating plants. And so that's all this map that you see here on the right. So we got all the possible dads of all the species. And then we collected seed from some of the possible moms, which of course, as you know, for most of these species are all the same because they have both male and female flowers. So we collected seeds. And then what we're doing then is Zach, who is a technician on campus at the University of Minnesota, he's growing up the seedlings from all of the moms. So what we can do, right, so this is an example of all the Rotibita seedlings in the greenhouse, and they're quite big. And so I'll just go back a second. So if we collect seeds from all of the possible moms, we can subtract the mother's DNA from the tissue we collect because we know who the mom is and where she's located. And then we can use basically maximum likelihood algorithms to figure out who the most likely dad is based on the remaining genetic contribution from the seedlings. And then we can measure that distance and we know how far the pollen moved. And so we're basically playing a giant game of Jerry Springer, who's the father. I feel like we're Jerry Springer of the prairie. And so we're doing that for 3 species. And so again, Zach is responsible for extracting the DNA from hundreds and hundreds of plants. So he deserves a lot of credit. And you know, it's a busy job, and we're working with a new facility on Minnesota's campus, and so they have been remarkably slow, unfortunately, about getting our sequences to us, and have made some mistakes. So we were supposed to have pollen dispersal results by now, and I was really excited to share them with you, but of course, we're just working a little bit behind schedule, but we should have results soon, hopefully, but we'll know how far pollinators are transferring pollen on the landscape really soon, hopefully, which is great. And so we focused on those 3 species. And then this year, with the help of 2 other great technicians, we're focusing on 5 more, because we promised LCCMAR we would do 8 species. So this summer has been really fun, because I've been here the whole year. I got to start after July, earlier than July, which is how it worked in the first year. And so I got to pick various species, and unfortunately not unfortunately, but we have a lot of species now that are pollinated by insects. And that their seeds move in various ways. So some have fluffy seeds that, like GM, where it has little hairs on it, and it can disperse far. Other seeds, this Oxytropis lambertii is a pod that is eaten by animals, likely. So we saw a lot of herbivory in the field, so they can travel far distances. And then Penstemon grandiflorus and Delphinium carolinum, the seed pods open and the seeds just disperse from there. And Artemisia frigida is really cool. It's the one I'm working on right now. I'm collecting the last seeds from it today after this meeting, and it's wind pollinated. So it's been really interesting to see how many seeds are actually being produced, and we're going to be able to compare pollen dispersal kernels to wind dispersed pollen kernels, and it looks like just, from collecting the seed, there's many fewer seeds on these Artemisia plants than there are on some of these insect pollinated plants. So I'm really interested to see the results from that. Yeah, so we're still working on that in the field, and we're going to be able to compare. We're struggling a little bit with the seed dispersal. It turned out to be, well, it's just tricky. You have to find seedlings in the field, and as you all understand, that can be really tricky. I don't know if you've ever looked for seedlings in a prairie, but it can be hard. I'm hoping that they burn bluestem prairie soon, so then it'll be easier to find them. But so we're sort of taking a pause on trying to figure out how far these species are moving by seed, because we can use some really nice models to estimate that idea, how far the seeds are moving by their various mechanisms. Right, so we know pollen. Sorry, I forgot these slides right here. We've been able to figure out pollen, and as soon as we can get our sequencing facilities going, that will be no problem. But what we're trying to figure out right now is the seeds. And so we sort of switched tactics now, and we're trying to see if we actually see evidence of in the landscape if these plants are actually moving by seed and colonizing. And to do that, I'm working on a project where these are my technicians from this year. They indicated they're both undergraduates or recently graduated, and they indicated they wanted to do a project. So Jessica and I had actually been talking about working on a project looking at spillover, thinking about how plants actually move in the landscape, and these 2 seemed really excited. So there's Katie Sperry on the left and Haley Hilfer in the middle. And they were really excited about trying to tackle this question. So this is a project that the 3 of us are working on in collaboration with Jessica Peterson, who's been awesome and really helpful resource, and Ian Lane, who's a pollinator guy, grad student at the University of Minnesota. And this is us in a sea of hordeum, which was the first day of our spillover sampling. But anyway, what we're trying to do is ask questions about this idea of spillover. So does the movement of native target species from one habitat to another, are we seeing evidence for this on the landscape? And so to do that, what we're doing is we're sampling reconstructed prairies that are adjacent to various sources. So to see if seeds from the source are moving into these reconstructed prairies. And so the best idea you can get of that is if this green prairie right here in the circle is a remnant prairie and it's adjacent to this red prairie that's reconstructed, are we seeing species from this remnant prairie moving into this reconstructed prairie, and how many and how often? And so basically, again, here's the remnant prairie, and then we're in the green square, we're collecting species richness. So we're getting out there, we're looking at how many species there are and what their abundances are, and then we're measuring the abundance of the species that were planted and also that weren't planted, all the species that are in these red squares and these transects away from this source, to see if we can get an idea of how far seeds might be moving. And to do that, so we're asking basically two questions. So one, is spillover happening? Are we seeing these native species moving into these reconstructed prairies that that weren't planted? So basically, we've gotten all of the seeding mixes for all of the reconstructed prairies that we've worked on, and we're going to subtract those species out from what we see, and then see if any have come from the sources. And to determine if spillover is actually happening, we're comparing this landscape. So we're saying we're looking at the difference between if this green square is a remnant prairie, if the source is a remnant prairie, or if it's an ag field. So that way we can get an idea of how are species moving, and if we see them, just because we see them in restored, or when we have a remnant prairie as a source, could that just be from the seed bank? Could that be, it was in the seed mix? And so we're doing that by comparing transects that are adjacent to remnant prairies versus agricultural sources. And then we're also looking at if we see species moving, which we definitely are, which is really exciting. We're interested in what kinds of species move best. So we're trying to compare this movement across various different movement groups. So are there certain species like our weeds or invaders? Are they moving? Or are these sources of habitat for, or a source population for weeds that can then move out? Or is it species that disperse by wind? Are they able to move more easily than species that move by animals, or et cetera? And so that's what we're comparing. Unfortunately, well, I wouldn't say unfortunately, but we're actually moving quite quickly on this project. But it is the responsibility of the students. They are taking this project on, so we're sort of at the mercy of when they have a chance to get the data cleaned up, and we're very close to that, but we're just finishing up now because this season has been a little wacky. So we're just finishing up the data cleanup right now, so we should know pretty soon. So if you're interested, feel free to contact me. I'd be happy to give you more information on what we find soon. And with that, I just want to say thank you to all of the technicians who have helped in the field. Jordan, Zach, Dylan, Haley, and Katie, you guys they were awesome. And then Tony Borman, Allison Wallace, and Brian Wissenden from University of Minnesota Moorhead, and they have been really wonderful. I've spent 2 summers now up in the Fargo-Moorhead area for 4 to almost 7 months both times. And it can feel lonely and far from home when you don't have a place to work or an office or any sort of equipment, so they've been really wonderful. And Matt Mecklenburg with the Nature Conservancy has been also really great and really helpful with any questions that I had. And so with that, I will definitely take your questions. I wish I had results for you, but hopefully soon. Maybe I can come back next year and talk to you guys again about this. So that's all I have for you.
Thanks, Lauren. I'm happy to pass on questions if anybody wants to, write in the chat, or if you just want to unmute yourself if you're capable of that. Either way, I have a question just as other people are kind of pondering this. One of the things that we've been thinking about at the DNR, but also across other agencies, is creating seed source zones. Do you feel like the results of your dispersal study are going to shed any light on questions about how far we should be moving seed for reconstructions?
Yeah, that's a good question, and I get that a lot. I would say yes and no, typical scientist answer, I guess, but yeah, we'll get some idea of how far these natural pollen and seed dispersal processes are happening, but the results are very small scale.
Right.
So yeah, I think we can get some ideas, but I would really hesitate to say, right, because the work we're doing is definitely on more of like a local population scale, and so, I mean, the work we find might just be that everything has to be within a very short distance, and that's just not very realistic, I think, when you're trying to get seed for all of the restorations that you guys are all trying to do. And so when we find out what their answers are, I'll be able to answer that better, but I think it's sort of a yes and no answer, unfortunately.
Sure. Yeah, that makes sense. Anybody else have any questions for Lauren? Oh, Phil wants to know, are any of your study sites being grazed?
No, well, okay, natural grazing from deer, and definitely we see a lot of
I think
deer
he's
grazing.
probably thinking about cattle.
Yeah, no big animals. In the spillover sites, we did go to one remnant that was grazed, and that was a little tricky. But then after that, we just pretty much avoided all sites that were grazed. But for the pollen dispersal, no.
Well, I'm happy to pass on Lauren's contact information if anybody has additional questions. If there aren't any other questions, next week we have Marissa Allering, a Prairie ecologist from the Nature Conservancy, to discuss her project on land management as an important driver for grassland bird abundance. Mary Beth Block will send out the LTT news and links email tomorrow, and it'll include the recording of this week and last week's webinars. So hope you can join us next week. Thanks, Lauren.
Yeah, thank you for having me.
See you later.